DNA replication. DNA replication begins at origin of replication. Enzyme helicase unwind the double helix strand of DNA by breaking hydrogen bond and separate two parental strand to become single strand, forming replication bubble. At each end of replication bubble is a replication fork. When parental strands of DNA are being unwound, both DNA strands act as template. Single strand binding proteins hold the separated parental strands apart while they act as template. SSB protein bind to the unwound DNA strands and stabilize the single-stranded DNA. The unwinding of double helix causes tighter twisting and strain ahead of replication fork. Topoisomerase helps relieve the overwinding strain ahead of the replication fork and relieve strain by breaking, swiveling, and rejoining DNA strand. Primase catalyze the addition of RNA nucleotides that complementary to the template to form RNA primer. This is because DNA polymerase cannot initiate the synthesis of new DNA strands. They can only add DNA nucleotides to the end of an existing chain that is base paired with the template strand. The existing chain referred to RNA primer. DNA polymerase 3 adds free ties to the exposed bases by specific base pairing complementary to DNA template at 3' of RNA primer. DNA polymerase 3 catalyzes the elongation of new DNA strand at replication fork only in 5-3 direction. Leading strand is a new DNA strand synthesized continuously along the template strand, which elongates towards the replication fork. Only one RNA primer is required for DNA polymerase 3 to synthesize the entire leading strand. Lagging strand is a new DNA strand synthesized discontinuously along the template strand, which elongates in the direction away from the replication fork. Lagging strand consists of short fragments called Okazaki fragments. More than one RNA primer is required. DNA polymerase. I removes the RNA primer and replace with DNA nucleotides. DNA ligase catalyze the joining of all Okazaki fragments by forming phosphodiester bond together to form a continuous strand. Two identical copies of the original DNA are produced.